All right, everyone, let's get started with today's session. Today we will cover more about segmentation. We touched base with it yesterday, but we're going to cover it more in depth here, and we're going to touch on image filtering. And this filtering is going to be useful both on the raw data images and sometimes on a post-segmented image. We'll see in instances where there's still a little noise remaining as well. Remember again, we are your local integrated cellular imaging core. Consultations and questions are always free, so don't hesitate to reach out to me, Hunter, or anyone else in our core facility that you have questions for with imaging, with experimental design, with data analysis. That's what we're here for. And we appreciate seeing you and helping feed your interests. All right, so the goal of today's workshops, we're going to cover different types of filters. We're going to apply them. We're going to go over what mechanically a filter is when it's manipulating an image and working with pixels. This is just going to be a touch on the more mathematics side of data analysis. I will not ask you to do any math. Don't worry, this is not a maths class. Ideally, we can show you how to use these tools in a number of situations, not so that you're just following a set of instructions I give you up here, but so you can look at it with more informed eyes and then adapt your solutions to the problems you have. And we will do a number of exercises like yesterday, just to cement that kind of understanding. For filters, as I mentioned, I'll introduce you to the theory behind them, how they actually operate behind the scenes, and then what that means later on. I'll also give you some example filters that are pretty useful and pretty widely used actually in bioimage analysis for removing noise or enhancing boundaries. And we'll see more what that means later on. So what is actually the goal of image segmentation? Well, we've got an image and we've got some fluorescence on it. What do we do with that? We fundamentally want to separate all of the signal that we don't care about. And that is background, noise, autofluorescence, everything which is not the thing we're actually looking for. And to do that, we use a process of segmentation. From that process, we can extract things like masks, which I talked about a little before. A mask is essentially just an outline of areas of signal in the image. You can see below, we have masked the cytoplasm of a very convoluted, geometrically complex cell. Now, that doesn't necessarily mean there aren't any structures inside the mask for us to look at, but as a first approach, this removes sections of the image where there's nothing of interest going on. And that's helpful, one, because it saves us time, and two, because it gets rid of noise that could complicate our analysis at the end. So, filtering. What is a filter? How does a filter work? A filter, and this goes for almost all the filters I know about, there may be some which operate differently, but for the ones I'm aware of, you use something like an identity kernel. And what is an identity kernel? Well, you can represent it this way. Basically, it is a range of pixels centered around the pixel that you want to filter for. And the way that pixel is modified depends on its neighbor pixels. The identity kernel here is a trivial case. All that means is if you've got this pixel, you're filtering it, that pixel will always be the same value. It's not changed. This is a conservative operation. That doesn't change any data inside. If you wanted to use another filter, then its kernel would apply to the central pixel as well, and then kind of spread out and take information from the adjoining pixels. We'll be able to see that in cases where we run a Gaussian filter, and I'll describe what that is when we get to it. But essentially, it means it looks at the central cell or a central pixel, looks at all the pixels around it, and then it modifies that central pixel based on its neighbors in that way. We can skip this, but for those who remember matrix multiplication, that is not how the kernel operates. It makes it very much more confusing. But we don't need this, we can skip this. So, some common filters, I use these almost daily. A Gaussian is a blurring filter. It uses a Gaussian distribution to smooth out an image. So what do I mean by that? If we open the nucleus-gray TIFF, we can see we've got very clear signal and nuclei. We can find the filters under process filters, and that has a lot of the basic ones. So we can select a Gaussian blur. Quick note, a filter alters your data. An image that is filtered is not the same image you started out with. So always keep your raw data and always keep track of what filters you apply and how you've applied them, because that information is very important at the very end when you're going over in your analysis and determining its accuracy. So with the Gaussian blur filter here, we can either use a sigma radius, which is relatively abstract, or scaled units. I find the sigma radius is generally useful enough, and we can check this box to preview it. Once we apply this filter though, we cannot unapply. This is a destructive process. So if we apply a radius of, say, 1 sigma, we see originally the detail inside of the nuclei gets a little more faded. And the higher the sigma we make, the less and less detail we preserve inside of the structure. And we also dissipate or distort the boundary, where before we had relatively clear boundaries, still not with homogeneous signal, but we can identify them pretty concretely. Here, they're washed out, they're spread out. The Gaussian blur filter is useful in instances where you have a very diffuse, noisy signal that's present in some locations, and you apply the filter so you can kind of smooth that into the background. The caveat, as I mentioned, is also that it destroys internal features of your structures and the boundaries as well. So what uses are any filters to me if they're constantly destroying my data? Well, they can be used in conjunction in order to only perform certain functions, to subtract certain kinds of noise and data. The median filter is in some ways the opposite of the Gaussian. It preserves boundaries. So if we were concerned that, let's say, this nucleus here was maybe a little diffuse around the edges, maybe the boundary is a little dim, and we're worried we might confuse that with the background when we're doing a segmentation, we can do a median filter. What a median filter does is it looks at, if you think of the identity kernel, you've got your central pixel, it looks at its neighbors. Whatever the most common value is, that's what it sets the central pixel to. What that means is it's a very good filter for preserving edges or even enhancing edges while removing the inside information here. We can see how even as we increase the radius, the boundary of the cell remains more or less the same. At 6 sigma with a Gaussian filter, we would have already washed out the signal. but it remains here. It is useful in instances where you may not have a homogeneous membrane. I see this sometimes in really high resolution images. The membrane would not have stained equally all around, so you have little gaps in it. And when you're trying to segment it, even though you know it's a cell you're segmenting, the algorithm might say, well, there's this little gap in here. And that could be for a number of reasons: lack of fluorophores, lack of signal, presence of noise. But if we want to create an accurate segmentation of the cell, we might want to run a median filter through it so that those gaps can then be closed. And what does that look like? Yeah. If we can see this darker edge here inside, it's unlikely, but it's possible that a segmentation algorithm would cut this part out. If we run a median filter on it, then that becomes less severe. The boundary is still preserved, and we have a more concrete structure we can then segment. And top hat, that is actually a combination of several filters, but they're used in conjunction with each other pretty effectively. So the top hat filter, in fact, filters it filters in opposition, or in the reverse, let's say. The larger the filtration value, or the filtration setting rather, here, the larger the radius, the larger the structure that will remain. So we can see at 1 pixel, we filtered out some of the background inside, but we've also damaged the structure. We've gotten rid of potential signal. We can zoom in here. Process, filter, top hat. And see that while, yes, we've gotten rid of the haze in the center, we've also gotten rid of potential signal and structure out on the edge. If we increase those pixels, we're still getting rid of the center, but we are preserving more of that other structure. And the top hat filter, very good at preserving those structures while also getting rid of that background haze. And this works for all sorts of structures. So this is definitely a filter I would keep in mind. We can see here as well. We've still retained the general structure. Some of this on the periphery, difficult to quantify, but we've definitely gotten rid of the haze here in the vicinity of the cell. So it's definitely very powerful, especially when you have some background noise to filter out in your data. Something I neglected to mention, the median filter is very good at dealing with this specific type of noise called salt and pepper noise or shot noise. That's the kind that, you can see here, gathers up punctates around the image, can definitely frustrate a lot of segmentation and analysis, but can be very easily removed with a median filter. Yeah. So why don't you go ahead First, does anyone have any questions so far? All right. Yeah. So go ahead and try this exercise here. Utilizing the Gaussian blur filter, pull up the nucleus tandem gram lab, and then top filter the resulting image. Try a few values, and we want to see the preservation of those structures and the elimination of the background noise. So for the next 10 minutes, give that a try. Hunter and I can answer any questions you have during. Oh, everyone's had a chance to play around with the filters, ask some really good questions. Hopefully you're in a better position to use them in the future. I'm sure you are. We're going to move into segmentation and quantitative analysis now. I'm going to go over the exercises we're going to do the rest of the session. I'm going to cover the concept of deconvolution briefly here because it's pertinent, but we won't be doing deconvolution in the class today or in the seminar today. So the overall process of image segmentation: design your experiment, get your image, process and filter it. Ideally, you don't, but if you have to, make sure you note how you did it. Threshold that image, separate your structures of interest from your background, then potentially create a binary mask. The nice thing about those is it's such a widespread thing that is used, a binary mask, that if you create it in Fiji and you save it as a TIFF, you can import it in a lot of other software to as a pre-segmented mask to inform it and to have it do a lot of other calculations. Cell Profiler is a big one. It's open source. It's used a lot for characterization of cells, geometry, intensity, etc. It can take in masks from other programs. Similarly, I'll show you how to use Stardust to segment an image and then save those ROIs as a binary mask that you can then later call either in Fiji to run calculations or in another program that could use it. And the last part, after we have all of our support components put together, we can analyze the data at the end. Best part. Yeah. We went over, analyze particles. We went over that the other day. We can get all sorts of information. So, touching upon deconvolution, what is deconvolution? Hand in hand with that concept is what is a point spread function. So, there is some ground truth object, some ground truth sample on your slide or in your well or fixed and cleared and on the lattice or on a light sheet microscope. There is an object. We are interrogating the structure of that object with photons of a certain wavelength to excite a certain fluorophore or whatever. That object we will never see as clearly as it exists because each of our systems has a point spread function. That is an intrinsic distortion to the light that passes through the microscope. Through the microscope to the camera, to your eye, through the monitor. So what deconvolution attempts to do is remove that point spread function from the microscope. You can do this a number of ways. You can either calculate a theoretical point spread function based on the NA objective, the kind of light you're using. All of this stuff feeds and informs it. Or you can measure a PSF by using a really small bead in a sample to calibrate the microscope. In either case, the result is you're trying to remove that PSF from the image. And ideally, when you do that, you have something that is closer to what the object physically looks like. There are a number of limitations and issues of in this process. We won't go over them here. Suffice it to say, if you hear the term deconvolution, that's what it's referring to. That's what it means. We have a licensed software here at the Core called Microvolution. You can use it to deconvolve images if they need that kind of treatment. A lot of the more modern microscopy softwares deconvolve in tandem with taking images. So this may be the last time you are exposed to deconvolution, because it'll be in the working path for imaging in the very near future. In fact, Hunter, correct me if I'm wrong, but our Stellaris here on floor 3 does decon while it's imaging. Okay. Yeah, I'm pretty sure it does. Mm-hmm. Yes. Yeah. Mm-hmm. Yeah, definitely. On our workstations, we've got microvolution if you all end up needing it. But as with any filtering or data treatment, what are the drawbacks? The issues with deconvolution is if you over deconvolve something, if you don't have a good idea of what your PSF is, if you don't have a good theoretical PSF, if you don't have those things, probably don't do decon. If you have to, these are some of the issues you might see. If you over deconvolve, you can start erasing signal. We see here between A and C, we've lost a secondary piece of information here. And this is a sample of a neuron, I believe. Similarly, we can enhance noise and amplify something called ringing, which is when you deconvolve along a structure, but you enhance some dark or bright structures because of the structure of what we're looking at here, you can end up with artifacts. So it can enhance an image, wipe away some noise, reveal new structure, but if it's not done correctly, it could introduce a lot of artifacts. It could make analyzing your data the rest of the time tricky. In fact, some of you asked me a question about if you go too low with the top hat filter, you start seeing these stars in the background of the image that you were looking at. And that's the kind of, computational artifact that can arise in some of these filters and deconvolutions. And it's something you should keep an eye out if you're ever using this tool. The material here is very good, very useful for later reference. But we can begin by, actually, before we begin, let's go ahead and take a break till 10:55. And then we can come back and move on to the actual practice of thresholding. All right, let's move on to the next section. Let us try segmenting based on what we learned yesterday. Where are we? Yeah, so I went over this briefly, but there are several different methods of thresholding. Let me show you where they are. If we go to adjust threshold, we can open this interface, and this lets us do it manually. From the dropdown, though, we can also access a set of other thresholding methods. And if we scroll to them, we can see slightly different behaviors. What we want is to find a threshold method that works well for our data, this data. Is this going to select the structures we're interested in? It's useful to know this, and these are by all means not the whole of thresholding methods out there. These are just the simplest available in Fiji. But say I really want to find a good thresholding method, but I don't want to create an image going through each of them one by one. Well, there is a function called auto threshold down here, and that lets you select a method, or you can try them all. Depends on your data, but here we are dealing with white objects on the black background. Conceivably, you can get data sets where the reverse is true. We don't need this, we don't need this. Can click OK, and it will take a moment and then generate this kind of image for us. Right? Is it working? Yeah. Okay. So it is working in the background. We have two issues here. For the intermodes method and the minimum threshold method, it could not converge after 1,000 iterations, which means those two are probably a bad choice for this image. One of my, and this is neither here nor there, one of my least favorite things about Fiji is how it does not let you know if it's still processing something in the background, always, at least. So sometimes, even if you don't see it doing anything, give it a little bit, it might just be taking longer. What I like to do is, on Windows, I'll open the task manager and check my CPU usage. If it's higher than normal, then I know Fiji's running, typically. But what do we have here? We have a montage of all the different thresholding methods, and we can look at them side by side, much easier, and decide if one or several give us good data or a good thresholding. So the default works pretty well. Huang and Huang 2 work pretty well. Intermodes and minimum don't work at all. ISO data is not as good. We can see we have a really kind of messy outer perimeter because we've chopped it up a bit aggressively here, where we see a more comprehensive cell in the others. So ISO data, not the worst, not the best. Li is also good. Max entropy, which is one I use very often, isn't as good for this data set. We can see it catches a lot of the dimmer noise around the perimeter. But let's use the default one. That seems to work well. Well, no, we'll use Swan. Yeah. Yes.
So sorry, will you just go over how you got the different versions of the threshold again? Because I know how to get one, but I don't know how to stack them together.
Good question. If we have our image, we go to process or image, adjust auto threshold. So we don't want the threshold function here. We want auto threshold down here.
You did try all
Yeah, try all.
white objects on black background.
White objects on black background. Yeah, everything else is unchecked. Something to note, it doesn't, there it is. So it recommends we work on an 8-bit image. They support 16-bit images, but they could take a while and it may crash. Oh, interesting. This is, in fact, a 16-bit image. So we saw how the process worked when it was 16-bit. Let's try it now as an 8-bit. Much faster. But as I mentioned yesterday, that can trim some of the brighter data away because we're casting it as a lower bit depth. Has everyone been able to get to the other threshold and run this? Anyone having issues still?
You typically always do the trial, like auto, construct auto, or auto threshold to start.
I typically do it when, like, the thresholding methods I usually go to aren't producing good results, then I'll go and trial. The issue we saw as I was running it is the larger the image and the higher the bit depth, the longer it can take to process. So if you're running it and it's taking forever, you can always crop out a section that has some representative noise and some representative signal, and then threshold that and take it as a baseline. Because if it works on the cropped segment, it should work on the whole image. All right. So that was the auto threshold function. We can, here's a question for everyone. How do we subtract background from our images? Say we have some hazy noise. Oh, we already cleaned that one up. We need to open the old one. Cropped composite, image, color. Split channels. If we wanted to subtract the background and get rid of this haze, what's the best way to do it? We could naively go into our settings and just subtract the 10 dimmest pixels or the top 30 pixel bins, which are dim from the histogram. But that is actively removing data from the image. Alternatively, we can use something like a rolling ball radius. We talked about it earlier, and we saw how it gets rid of background even inside structures while preserving the structures themselves. So why would a math subtraction, why is that not great? Because if you recall back to day one, when we do something like that, we're changing the histogram. We are altering the image. And we can see that in the examples up here. If we do a rolled background subtraction, the two histograms are different. We have gotten rid of some of the background noise here. And we can see that in the image, the central areas of the cells are now closer to the background because we've removed that noise. However, the general structure of their histograms still remains the same. So we can be confident that this did remove noise, but it preserved our signal. If we were just to subtract the bottom 30 dimmest instances to get rid of that haze. We will get rid of the haze, but what we're also doing, and what we're doing indiscriminately, is subtracting from the structure as well. We're subtracting that brightness from the whole image, and that really changes the histogram at the end. So right now we can't, I mean, they're similar, but they're not that similar. We've lost a lot of the structures here, near zero. And that kind of loss can put our conclusions into question. Does this make sense? Let me rephrase. Does anyone not understand? Okay. After that background subtraction, we can also do our thresholding at that point. And we can see for the same thresholding methods, a generally better result after the rolling ball radius background subtraction versus before, because that haze in the side of the cells is no longer confusing. It's no longer complicating the image for the thresholding algorithm. So let us do today what we did last session. Open up the nucleus gray, that TIFF, and using everything we've learned so far, segment it into those 8 nuclei and populate the ROI manager with them. So go ahead and take 5-10 minutes to do that. Let us know if you need any assistance. Perfect. So we all got to our mask of nuclei. Did everyone get to the point where they ran the particle analyzer, or who didn't get to that point? Okay, and we all counted 8, right? Excellent. But let's say now we've populated our ROI. What do we want now? We want the actual measurements, the actual statistics. So at our ROI manager, if we run it with this selected, we will populate the ROI manager. We will populate our results as well initially. Now, a bit on the ROI manager. If we have an image selected, and let's actually take up the base image. We can look at which fit is where. And as you're segmenting data, I would recommend you do this a couple of times, because we see the default looked good, but we are missing the outer edge of this guy here. We'll go to the next one. Yeah, this is perhaps too conservative a fit because we have some signal outside of it. What about the others? This one is pretty good, but again, we are under capturing it. This one's close. But, oh, this one's really poor. So those are the kinds of things to watch out for. At any point now, we can select these individually, watch them load on the screen. We could show them all at once, along with their labels, if we wanted. We can measure. If we select one, we can measure, and its measurements get added to the end. We can rename them if we wanted to investigate them later. And most importantly, we can save these ROIs. So if we click Control All or I should be able to select them all. Yeah, Control Alt to select all of them. We can go to More and then click Save. And what that does is that saves us all of those ROIs. So if we were to close the ROI manager later on and we wanted those exact ROIs later, we could just open them again. Very useful. Now, from yesterday, we can set the measurements we want. Area, mean gray value, min and max. Let's say we want the centroid as well. That's good. That's enough. If we close we can also save these as a comma separated value file if we wanted. We can close it. Then we can populate it with the measurements we want. So click on one, click measure. We've got the area, mean, intensity, min and max, X, Y, and Z. So this is one of the biggest utilities of creating a mask with a thresholding method, because we can then go from just knowing where the different structures are to actually characterizing them based on our data. And the mask we can then feed into other programs if we needed to. So it's very powerful. And once we have it done, we can do a lot with it. So I will show you some of that. I briefly introduced the Biovoxel toolbox yesterday. If people have not, go ahead and download it. We can go to Update, Manage Update Sites, and then select Biovoxel here. Install it, reset PG. After we reset it, we'll come to this More Tools arrow at the far end, and it will show up here as the Biovoxel toolbox. Now, if we have is anyone in the process of downloading it right now? The Biovoxel toolbox? Okay, I'll give you a few minutes. Once you've been able to download and install the Biovoxel plugin, go on and try to complete this exercise. We'll be using it for this purpose. You can take and use the mask we've just created from the nucleus-gray.tiff file. What we want to do is have Biovoxel give us a shape descriptor map. So if we select our mask, we can go and select the shape descriptor maps and fill out this set, these settings here. Our particle colors are white because that's our segmentation mask. Do watershed first. This is for structures that might be very close to each other, two cells that are right next to each other. And the watershed will attempt to split up such clumped objects. So it is useful for one instance here, I'm sure. Yeah, if cells were touching in this way, the watershed algorithm will attempt to, create a break in between them at the most logical place, which is not always what you would think is the most logical place. But we don't need that here because none of our structures are touching. None of them are next to each other. Do we want to exclude edge particles? And edge particles is anything on the edge of the image. Say we had a half cell here falling off the edge. Do we want to exclude it or not? It depends. If you are trying to capture the entire geometry of the cell, then you wouldn't want that cell because you don't see all of it. You only see a part. If you were just trying to count the number of cells in the image, that could be acceptable if you had enough of it left to be sure that, yeah, that's a cell, that's not contamination or something else entirely. Include holes is sometimes you will segment cytoplasm instead of nucleus, and you have a shape and there would be a hole in it. Do you want that hole included or do you want it filled in? Again, it depends on circumstances. In certain instances, you do want that hole filled in because you're looking for a total area or volume of the cell. Sometimes you're looking for only the area or intensity of the cytoplasm, not the nucleus, not the membrane, in which case you would want to actually include the holes in that analysis. We want to show a calibration bar and enable interactive plots. So we can pick an LUT you like. Fire is generally pretty good. And we get the result, which is a stack that begins with our mask and then proceeds to the other measurements. Now, we've got area, perimeter, max farad diameter, all sorts of spatial statistics, spatial and geometric statistics, and we see they're color-coded by the value of that statistic. We can reference those here. So go ahead and do that again after you do it the first time. Do it to the crop composite mask. TIFF. Yeah. And that mask, let me open one just so you can all see it. When I talk about water shedding, this is the kind of structure that we've used water shedding on. Because the structures are so large and geometrically complex, the thresholding doesn't always separate them. So the watershed algorithm looks to find the barriers between those two, between the number of structures, and segment them just a little bit, just so they're not contiguous. So you can then use this as a mask somewhere else. But we can move on to segmenting nuclei with background issues now. So we've had the easy section done. Clear signal, no background. Now, what about things with both signal and background noise? Well, let's tackle this one. This file, nucleus tanim.gram lab. Let's go ahead and open that one up. There is quite a bit of noise in the background, and our structures don't always seem to be quite in focus. These two are also very close together. So we need to be careful, and we need to find a way to get rid of the noise. In this instance, we can try a threshold first. Modes. The first couple seem complicated. So we can go to auto threshold, try them all, and see if there's a good middle ground. It doesn't quite give us a good fit. ISO data. Turn-ons. So IJ ISO data captures most of the signal we're interested in, some of the background noise, and not a lot of the haze from the structures themselves. All right, we can click apply. So now we have a thresholded image and a mask image, but we've still got these structures in the background. So how can we filter them out? We can filter them with a number of ways, actually. We can go and utilize our particle analyzer and then filter by size. That way we'll get the ROIs of all of our cells. So let's do a little bit of measurement. And seeing what the size of our structure is. So let's go a long way. Up here we can see a length of 41 pixels. And this length is in pixels because this image doesn't have a scale. So we want to catch structures which are 41 pixels across, 32 pixels across, okay. But what are the really small ones? What's their area? They're about with height 5. Area 21 pixels. What's the biggest we can find? 86. So if these are all signal and this is noise, 210. We want to run the analyze particles function, and we want to say, what were these values? I was just looking at them. 80 to infinity. We don't have an upper limit. Our structures can't be too big in this instance. Circularity we don't care about. Clear results, display results, add to manager. Save for measurements, don't save. So now we have all of our structures and some of our noise. We can select only the ROIs of interest. That is not this one, 11 or 9. We can delete those. And now we have an ROI manager just filled with the ROIs of our own cells of interest. But is there another way we could do it? There is. If the filters, there we are. If the noise is small enough, we can get rid of it with a median filter. At this resolution, the really small bits of noise behave like punctum, like salt and pepper noise. So we can get rid of them with an appropriately large median filter. So at 3, it gets rid of a lot of the smaller noise. With, let's see, 10, it gets rid of almost all of the noise. However, we can see it begins to eat away at the edge of the cells. So if we were to apply it right now, this would be an instance of overfiltering. But we are satisfied with something like this, where we get rid of almost everything, just 3 bits left. What else can we do? Since we have the ROIs, since we've already filtered the ROIs for the components we want, we can create a new image. If you go to File, New Image, call it what we want now is to create a new image of the same size as the old image, because what we're going to do is we're going to take those ROIs and then draw the masks in the new empty image, instead of filtering this until we get rid of all of the small little puncta in there. So the old image is 614 by 532 pixels, because it's only one slice, okay. Fill it with black, because that is what no signal looks like. We'll click OK. We have a pure black image. If we select our ROIs, then we can fill the image with those ROIs, and then in effect we're creating a new mask image from this. So from the ROI, we can select an ROI. We can go to Edit and Fill, but first we want to make sure we're going to fill it with the right color. So we want to, if we right-click this, we want white and black, I believe. And that's going to make our fill color white. So we come in and we press Fill. Fill or Control F. And we can do that for every mask. And then that's our mask. We can save it, make sure it's there. But there's still the issue of these two structures. Now these cells were very close to each other. And despite all of our thresholding and filtering, they're still reading here as one object. At this point, we can apply something like the watershed. That's just a function. You call it, you don't have to provide any arguments, and it will look at all the structures and split them if they look like they're touching. And that's it. We've got our mask, we've got our closed cells that have now been watersheded apart, so we can tell that they're two distinct creatures. And now we can take a look at these shape descriptor maps. Particle color is white. We could have done watershed first. Let's see. Doesn't work. We want to show the calibration bar. We don't want to direct plots fire. Okay. And we get the same measurements we were able to get before: area, XYZ, etc. And the shape descriptor maps here for this segmented image. Mhm. Is anyone having issues following along? Any questions?
How did you threshold by object size?
Say again?
How did you threshold by object size? Like you measured the particle size?
Oh, okay, yeah. So what I did was back when there was noise, yeah, I went around either with the line tool or with one of the other tools just to get a rough estimation. So I did a brush to select this. I pressed M to measure, and what that will give me is the area in pixels because this image doesn't have a scale. So once I know the area of the biggest thing I don't want to count, I then go to, either the extended particle analyzer from Biovoxel, which I really like, or the analyze particles function of Fiji. We can then just specify that here. So with an area of like 80, I filtered by that. So I said 80 to infinity. Anything bigger than 80 is something we want. That's how I set up the filter. That answers the question? Yeah. Okay.
So, okay. How does it? It looks like there's some really small particles on screen that it's selecting. Looks like it's selecting. See how when I click on this one, you see it right there. Not sure why it's only showing. Why don't the highlights on and then does a strange change of size? So it looks like it's selecting. So tell which ones get selected by which one gets pressed. I haven't seen that. White background here. We try that. Also try this. Process, binary, options. Now let's try process. Okay. Change the with options and just turn off white background. This is a white background. Okay. It shouldn't matter because the values are 0 and 255. Yeah. Okay. Great, so now when you click on one, it just goes and changes it. Perfect. Now that's what I'll tell people to do. We had some issues with the result.
All right, it looks like everyone has more or less proceeded through the segmentation and particle analyze portion for today. Great job, everyone. Let's take a 20-30 minute break right now. People can go out and have lunch, and when we return, we will continue with a slightly more involved application of segmentation, where we're actually just looking at the membrane intensity around a cell. So that's going to be the fun process of masking cells regularly and then making them bigger and then calculating what we think the membrane should be. All right, everyone, hopefully we've all had a restful little break. We can come back to the next to last exercise today, which is applying the knowledge we have cultivated in this environment to now analyze the intensity, but of a cell membrane. So how can we do that? First, we will begin by opening up the nuclear membrane image. Fiji, let me close on all these windows. Let's see. So we have two channels here. We have a central channel and we have a more diffuse membrane channel. How can we see that more clearly? We can go to make composite or no, channels tool. There we are. And toggle on and off the various channels. This is the one we will use to create our cell perimeter. And channel 2 is the one we will actually apply the cell membrane to as a mask in order to calculate the intensity. It's important to do that correctly here because we see the signal on the green channel isn't just localized at the membrane. Ideally, it would be. It would only stain the thing we wanted to stain, but it instead diffuses out of the cell and inside of the cell. So in this case, it's very important that we get the membrane just right. So how can we start doing that? Well, we will begin by first duplicating the nuclear channel. So which channel? Channel 1, I believe, is the nuclear channel. Oh, yeah, channel 1. So we go to channel 1. We can color channels tool. We set this to, we set this to color. So we've got the two channels here. We want to duplicate just this one. Yes. So when you open this image, it is, I believe, a composite image. I opened the channels tool with a drop down, went from composite to color. Right. So from here, we can go to edit or image duplicate. I did this once before yesterday, but to remind all of you, duplicate can duplicate an entire stack, an entire hyperstack. We can make a duplicate of this, but it's only 2 channels, and we only want 1 channel. So we'll uncheck duplicate hyperstack and select, we'll select channel 1, which is the channel we want, and at the end we get a image just of that channel. Great, so what do we do with this? We can subtract background of 50, or we can try median filter with a Gaussian blur. What do we need to filter on this image? Because it looks pretty clean. Let's see. Right now, it is in the more or less the auto brightness contrast setting. If we take down the maximum value, we will, of course, oversaturate our structures here visually, but we'll also enhance whatever really dim background there is. And it is very dim. And that, I do that just to get a sense of what my data looks like, because looking at it just however you pulled it up can hide some information. If the brightness and contrast settings are off, you can get the impression that, say at this level, you're missing a signal here, but you have it elsewhere. Well, that's not quite true, because if you modify it, you can more clearly see it. So play around with it, especially when you're looking at new data that you haven't looked at before, because it can be informative. But what do we see here? Well, we see these are pretty complete, but some of the other ones have that characteristic that I was mentioning earlier. That is, their cell boundary isn't necessarily homogeneously illuminated. We have some brighter areas and some dimmer areas. And what that means is that if we attempted to threshold it now, we may not get the entire cell. You can see up here, we're only getting parts of it. We're leaving a lot of the perimeter unthresholded. So we can skip through them, look to see which one gives a good fit, but there's actually something else we can do as well before we get to the thresholding, and that is we can filter it. So when I mentioned previously the Gaussian filter and the median filter, this is an instance where the Gaussian filter would be good to use. Because while we want to preserve the edge, we also want to kind of wash away the central non-homogeneity of the cells. Because at this point, all we're looking for is to get the outermost perimeter. But instead, actually, there's something else I mentioned. If we were to use the median filter, that would preserve the edge, but also wash away some of the non-homogeneity on the inside. So let's try that instead. Oops. Under filters, median. You can see it's 6, it might be over much, about 5. That seems better. We are getting rid of all of the internal details, so kind of solidifying the perimeter a little bit, especially compared to the background. So we'll click OK, we'll zoom in, we'll say, they are cleaner on the inside, the perimeter is preserved. Perfect. At this point, we can now threshold this image. But just in case, Let me duplicate it in case we make a mistake and we want to reference it again. We can go and adjust threshold. And compared to the first time, we can see a lot of the thresholds are now fitting more clearly to the whole cell. This one was being captured in pieces. Right now it's being captured as a whole cell. Okay. Once we're happy, we can apply it. And that creates our mask. Now, there's one issue. We can see it up here in the corner. These two are distinct cells, but they've been thresholded to be a single cell. So we can go and run the watershed algorithm, process binary watershed, and that will separate these two. It will not change the rest of the structures we see. And now at this point, we can generate the ROIs. So we can do that either with the extended particle analyzer or the regular particle analyzer. We want everything, so we're not going to filter by size. We're not going to filter by circularity because everything here is a signal we want. We want to add them to the manager. Exclude edges, exclude holes. We do have one cell, in fact, up here at the edge of the screen. If we look at our original image, do we have a cell up there? We do. It's very dim, but it is here in both channels. So, who here thinks we can ignore this cell in the corner? Raise your hand. Who thinks we can keep it? Raise your hand. Okay. We shouldn't keep it. Why shouldn't we keep it? Well, we have a clear component of it here. We can see it, we can fit it, we can threshold it. However, once the particle analyzer is at the edge, it's going to have to complete that shape. It can't analyze just an open curve, so it will draw a line behind it. Because that line would go through the body of the cell, we can't really integrate the membrane intensity because we'll end up integrating a bunch that goes through the bulk of it. So yes, we do want to get rid of that one. We should not count it because the way in which we'll end up measuring it is not going to give out good data. So back to analyze particles, 0 to infinity, circulating masks, exclude on edges. Perfect. We don't care about this. We have no holes. We don't want an overlay or a composite ROI. We do want to add them to the manager. So once we click OK, we see our ROI manager filled out with all of the structures here. And we have a nice mask. But what do we want? We don't want the central bulk of the cell. We just want the membrane. So now we fit the central bulk. How can we calculate the membrane? Well, it's a good thing we are already at the masking portion because that makes it a bit easier. So let us duplicate the image just in case. We've got this image, we've got our cells. How can we make them larger or smaller? Well, if we go to process math, right? No, binary. Yeah, process binary. We have functions here called erode and dilate. And what do these functions do? Erode basically for everything in the image that is a mask. Any area which is white will be reduced by 1 pixel. Dilate is the opposite. Any area which has a white pixel will be expanded by an additional pixel or more. You can set it once we open those up. So let's zoom in to one of our structures so we can see the process. Let's see, which guy is that? Show all labels is 4. So this one is 4. We can see the perimeter of the ROI. We can then go to process binary dilate, right? Process binary. Yeah, so erode works. Dilate works. Okay, there we are. So yeah, back 1. And if we use it, we see they all grow by 1 pixel. Similarly, we can, it wasn't dilate, it was erode them. And then they'll get even smaller. But this is for the outermost structure of the cell. So what do we need to subtract from that to get the membrane? Well, we can do the erode function here. Then these are just a little smaller. And once we have these smaller, we can then perform math on the image. How can we do that? Under image, I believe, process math. We can add and subtract, multiply, divide, and a lot more esoteric manipulations. But this image is the smaller internal area. This is the larger area that encompasses the edge of the signal. So we can subtract the second from the first. So how would we do that? We can go to process, math. Oh, shoot. Was that just a regular calculator?
Just on process.
Yeah.
You would already recommend an image calculator and load batch and upload some tracks back. Go load.
Oh, there we are. Yes. Thank you, Hunter.
Yeah.
When I said I am still getting lost in the Fiji menus, this is exactly what I mean. So, image 1, that is this one. Subtract image 2, that is this one. Create a new window, image 32-bit float. This should not be necessary for most situations. But it may be essential for some plugins that require very high precision. But once we click OK, we are left with just these guys, just the perimeters. Now, the only thing left to do is to see if they fit around the nuclei correctly, and if we can create a particle analyzer of them. And we can. We see each one here. And we can then go back to our first image. And show them all. But what do we see? That the calculation we made doesn't quite fit along the perimeter. In both channels. It fits this guy pretty well. This guy, but it's not great for all of them. Well, that is a good start. What are some issues with this? Well, if we erode too much or too little, we can overcount or undercount. That is what would happen if, say, we, it wasn't erode, if we dilated the mask twice or 3 times, we'd get a much thicker membrane. And it would also be capturing data we may not have wanted. Yeah, image calculator. But who hasn't been able to get to this point? Does anyone have questions? Take a bit longer to work on it. And while we're walking around, helping everyone, go ahead and get the measurements from the original channel. The original channel.
The nice thing about this is that it also allows you to make so like if you want the outline So have you already done the maximum filter? Okay, then let's go. So let's go to process filters. So here, watch the edges of this. You're gonna get a little bit whiter. And so here, let me close these things. Let's go ahead and close the ROI. Just make things a little easier. And so then is we have now this version of the image and this version of the image. So here, a mask of this is this big. All right. So let's do this. Let's go to process image calculator. Oh yeah. Okay. This one has 3 ones. No. This okay. Tracks. This is way easier. So now I'm gonna tell you if you get this. It took maybe yeah. Yeah. I don't think okay, then maybe this is just another way to do it. But like but this is still how you do it. Like it's really bad. Yeah, you take one image, you hit, and then run the maximum function on one. Basically, what the maximum function is doing is like within 2 pixels, which of those 2 pixels is the higher value, and then set both of them to that value. And so if you think about what that would mean on a hard edge, where one is increases their work by 1 pixel. So that's why when we go back and subtract the original image, we get these outlines. Yeah, so just give that a shot, make sure you get good at that. And then in general, I think what helps is to close out those images on image J as you don't need them, like save them and get rid of them. Because a lot of times what can produce issues is when image J gets confused as to what image you're trying to analyze. So give that a shot. Yeah, of course.
All right, it looks like almost everyone has gone through and finished the process. We are reaching the end of our time for today, so I will quickly go through the last part of the presentation. We'll talk about Stardust, which I mentioned briefly yesterday, but is a very powerful machine learning tool for Fiji, and it does a very good job segmenting images like what we just spent half an hour segmenting by hand, but automatically. So it is a UNet type of CNN deep learning model. What that means is they have a model and they have trained it on a lot of annotated data. So because we're going to talk a little bit more about this tomorrow, I'm not going to discuss it as much now, but essentially, if you have a set of annotated images, labeled images, and what those words mean is annotated and labeled, is a human has gone, looked at them and said, these are definitely the structures that exist in this image. Those are fed to the model, and then the model extracts the patterns that it sees in those images. So a well-labeled image generally trains a model that can effectively distinguish structures. If the image is poorly labeled, you're going to have a bad model. And this is more of a tangential story, but years ago, one of the experiments that they were running with machine learning was to have a computer look at images of tumors from hospital patients. They wanted to look at the tumors because they were hoping, an image analysis software that is built on an AI model can identify them without the same preconceptions humans might. So we want to train the model to look at these images of tumors and identify which ones are cancerous, and that way it can assist doctors. Very good. Good idea. They tried it out and they found out that what the model was actually picking up in each of the images wasn't the tumor, it was the ruler present next to a cancerous tumor. So because the ruler was always there for a cancerous sample, but not for a non-cancerous sample, that was the easiest pattern for it to pick up on, and that's how it identified whether it was a benign tumor or cancerous tumor. So what is the point of that story? Just to say that if you have a bad labeled image, you're going to have a bad sample or a bad output. And a machine learning algorithm isn't necessarily better than a human operator, especially at the edges of applicability. We'll skip this. I can go over more of it tomorrow. This is just a distinction between semantic segmentation and instance segmentation. Semantic segmentation is something like coming into this room and differentiating people from furniture. All of you are people, there's some furniture, we know those two groups. Instance segmentation is I actually go around and I learn each of your names. That way, each person is not furniture and also identified by a certain name. That's the distinction. So what we did with the segmentation by hand is first we did a semantic segmentation where we thresholded the image and we only had the nuclei. Then the particle analyzer helped us do a instant segmentation, because then we differentiated those nuclei from each other, and we got we got areas, intensities, that kind of stuff. Stardust, very powerful, only works on things with this general geometry, so closed convex polygons. Yeah, think cell nuclei. So this was originally an exercise, but I don't think we'll have time to finish it all. So I'm just going to show you how we can do some of what we did today much, much faster with this plugin. We have a complex image here. That is this one? No, it's this one. I might not have it on here. Give me a second. Oh, we can work on this one anyway. Yeah. Is anyone, does anyone not have Stardust installed for this? So you can find it under plugins, Stardust 2D. They are working on a 3D implementation, but I don't recall how far along it is. The model we're using is the fluorescence nuclei model here. These sliders are what inform some of the parameters of the model. Of specific interest are the overlap threshold and the probability score threshold here. What Stardust can do is, based on the geometry of the structures it's fitting, it can extend one structure underneath another if it sees that the structure is potentially two cells on top of each other. So it's really powerful for that kind of thing. It isn't always necessary. In this instance, none of the cells are overlapping each other, but it can be useful in other more complex images. This basically controls how likely the software is to identify a certain incomplete cell or nucleus as being overlapped with another. Now, what are the numbers you should set these to? Again, it depends on your data. When I'm using this, typically I will use it several times on the same image to see if I'm going in the right direction or the wrong direction. So it does take a bit of fiddling and playing around. The more you use it, the more you'll come to better understand what ranges tend to work for what kind of data sets. But fundamentally, you're gonna have to play around with it. There's no good way a priori to say, oh, this is gonna be around the value you're gonna wanna set it at. ROI positions are automatic. This does populate your ROI manager with the structures. So if you already have structures or ROIs in your manager that you want to save, go ahead and save them, clear it out. And we can see it right here. We can have it populate the ROI manager, only label the images or do both. We want to do both. So we click OK, and we see the segmented image. Because we didn't filter it a priori, we have all these punctated structures, but we also have our cells of interest. We show all and label. Yeah. We can take those ROIs to our image of interest and we can see how it segmented them. So right now we see on most of the cells, it did a good job. Some of them, like these two here, it over segmented. So these two cells are now 4. it got that one pretty well and that one pretty well. But once we have these ROIs, we can do the same thing basically. So let's start again. Let's filter the image to get rid of some of that noise, and then try and do the cell membrane calculation again. So we've got it here. Let's filter. Process filter. We want to filter these ROIs, so we can do analyze, particles, and filter by size. But what is the size of the ones we want to get rid of? Area of 84 pixels. It's the same image, so it's about the same area. 147. What's the smallest large structure we want to keep? 330. Okay, so analyze particles. We can say 200 to infinity. That should get rid of all the small ones. Do we want to add them to the manager? Yes, we do. We also want to clean up the manager. Yes. I got too excited. I skipped the, what did I skip? Thresholding. But actually, we don't need to because we used start this. That's why. What am I missing? Mhm. Yeah, once we threshold the image, we can threshold it relatively easily because there is no signal in the dark areas. So that way we can filter out the smaller punctated noise. But I would recommend that everyone give it a try sometime. It's free. You can download it from the Fiji update sites. At least as a first run for segmenting some wild images like this, it's definitely a good choice. I don't seem to have that image. Does anyone have questions on this subject so far? Is everyone able to get the macro or the plugin to run? Has anyone encountered issues with that? What's the issues?
It's saying that I don't have another plugin, and when I look for that, it's not there. CBSD.
You have to download it the same way you download Stardust. The URL.
I don't see it in the. Yeah, so does it prompt you to download TensorFlow? I would say just download the latest version of TensorFlow. Just things over here. Wasn't so you tried to update Surface and it said sorry you don't have TensorFlow. If it doesn't work well from there, also you can try going to the update list just like you did for Surface. And then yeah, sorry for my long I just made a note of stuff I gotta do. Sometimes it does. Alright, well. Maybe next time.
Gave up.
It was just like. Yeah.
Using that tomorrow.
So, are we gonna start our stuff tomorrow? We should have some time tomorrow as well if you want to try something from the other 2 days of training stuff. And then if you still want to try, we can do a consultation and just email us. Yep. Yeah, perfect. Hey, so for the changes to the sorry to interrupt you. For the changes to the presentation, you made those directly to the one you already showed. Oh, sure. If we don't cover the tomorrow, that'd be the consultation.
Okay, cool, yeah. I messed around with them a little bit and I think I just have like a few pieces of, lines that aren't in there that I cannot quite do what I want.
Yeah, usually with writing macros or using the record function, the thing that trips me up is I had managed to be able to tell which images was picked to do it.
Yes, exactly.
And so I think I actually ran into that exact thing when I read my thesis.
Yeah.
But yeah,
That's what I've been running.
basically the function I